Welcome back, my dear students. Today we are going to explore the biochemistry of monosaccharides, which is the simplest and most fundamental unit of carbohydrates. These tiny molecules are the building blocks of life's energy currency, and it serves as the primary source of fuel for our cells. But the monosaccharides are not just about energy. They also play crucial roles in structure, metabolism, and even cellular communication. Let's dive in and uncover why these simple sugars are anything but they are very simple. আমার লাস্ট ক্লাসে আমি একদম বেসিক ইন্ট্রোডাকশন টু কার্বোহাইড্রেট বলতে অ্যাকচুয়ালি কি বোঝায় এবং কার্বোহাইড্রেটের ক্লাসিফিকেশন, কত রকম টাইপের কার্বোহাইড্রেট হতে পারে এবং আমাদের বায়োলজিক্যাল সিস্টেমের মধ্যে কার্বোহাইড্রেট কি কাজ করে সেটা নিয়ে আমরা বলেছি। আজকে আমরা সেই এক ধরনের কার্বোহাইড্রেট, সবচেয়ে সিম্পলেস্ট কার্বোহাইড্রেট যেটা, সেটা হলো মনোস্যাকারাইড। সেই মনোস্যাকারাইডের বায়োকেমিস্ট্রি নিয়ে আমরা পড়বো। মনোস্যাকারাইডস আর বেসিক্যালি আমরা জানি দে আর সিম্পল সুগার্স দ্যাট কনস্টিটিউট দ্য বিল্ডিং ব্লকস অফ এ মোর কমপ্লেক্স ফর্মস অফ sugars like oligosaccharide or polysaccharides. The examples of monosaccharides include glucose, fructose and galactose. Monosaccharides consist of a single polyhydroxyaldehyde or ketone unit. So monosaccharide consists of a single polyhydroxy, that means which have more than one hydroxyl group and either a aldehyde group or ketone. They are the simplest sugar and they cannot be hydrolyzed into simpler forms. Monosaccharides গুলো এতই simple and they are the simplest sugar, so they cannot be hydrolyzed into simpler forms. Properties: monosaccharides are colorless, crystalline solids. They are freely soluble in water, but they are insoluble in nonpolar solvents. The most abundant monosaccharide is D-glucose. So the examples of monosaccharides are galactose, fructose, glucose, deoxyribose and ribose. These are the cyclic forms of monosaccharides and these are the linear chains of monosaccharides like D-glucose, D-mannose and D-fructose. Classification of monosaccharides by the number of carbon atoms. If the monosaccharides consist of 3 carbons, so they are called as trioses. They can be all the monosaccharides can be containing aldehyde group containing monosaccharide, or they can be ketone group containing monosaccharides. If the 3 carbon monosaccharides containing aldehyde group, they are called as aldotriose. The example is glyceraldehyde. The keto group containing monosaccharide is ketotriose. The example is dihydroxyacetone. Tetroses, which is the 4 carbon compound. The compound with aldehyde group, tetroses with aldehyde group, is called as aldotetrose, the example of which is erythrose, and the keto group containing tetrose is called as ketotetrose. The example of ketotetrose is erythrulose. Similarly, aldopentose, it is the example is ribose, and ribulose is the example of ketopentose. They are both the ribose and ribulose are the pentoses, and they are respectively aldopentose and ketopentose. Hexoses are the 6 carbon containing monosaccharides. If aldehyde groups are present in them, they are called as aldohexose, such as glucose, and if ketone group is present in the 6 carbon monosaccharides, they are called as ketohexoses. So the example of ketohexose is fructose. Similarly, 7 carbon monosaccharide is heptoses. Aldoheptoses are L-glycero-D-manoheptose, and the example of ketoheptose is the pseudoheptulose. Octoses, which are the 8 carbon monosaccharides. If aldehyde group is present, they are called as aldooctose, and keto group is present, then it is called as ketooctose. So the example of aldooctose is aldooctose, and the example of ketooctose is octulose. So the monosaccharides are the simplest form of carbohydrates that cannot be hydrolyzed into smaller carbohydrates. The examples of monosaccharides are glucose, fructose and galactose. Their general formula is CH2O whole n, where n number of n is 3 to 10. These are the classification of monosaccharides on the basis of carbon atom, and these are the classification of monosaccharides on the basis of their functional group, either aldehyde group or ketone group. It can be respectively aldoses and ketoses. So all monosaccharides are reducing sugar, we know. Glucose and fructose are isomers. They are same formula and different structure. Ribose is a component of ATP and RNA. Deoxyribose is the sugar in DNA. Important monosaccharides: glucose, its formula is aldohexose. Fructose, it is ketohexose. Galactose, it is aldohectose. Ribose is aldopentose, and deoxyribose is also aldopentose. Monosaccharides are basically instant source of energy, especially glucose. They are the building blocks of nucleic acids like ribose in RNA. They participate in cell recognition and signaling mechanisms. Now we should study the chirality and isomerism of the monosaccharides. All monosaccharides except dihydroxyacetone contain at least one chiral carbon. So they show optical isomerism. So first we need to know the diastereomer. Diastereomer are the type of stereoisomer or three-dimensional isomer that are not mirror image and not superimposable to each other. Examples of diastereomers are given in the diagram here. D-glucose and D-ultrose are the diastereomer. They are not mirror imaged and not non-superimposable due to the presence of the OH group in both carbon number 2 and carbon number 3. Now we should know about enantiomer. Here chiral molecules that are not superimposable mirror images. A molecule with n chiral centers has 2 to the power n stereoisomer according to Van 't Hoff's rule. We know in glyceraldehyde there is one chiral carbon, so it should have two isomers, and aldohexoses which has four number of chiral carbon, so it has 16 isomers. So D-glucose and L-glucose are the enantiomers of each other. Epimer, sugars when differing only at one chiral carbon, examples are D-glucose and D-mannose, they differ at C2, and D-glucose and D-galactose, they differ at the fourth carbon. Epimers, the two sugars that different only in the configuration around one carbon atom, is called as epimer. D-glucose and D-mannose, which differ only in the stereochemistry at C2, are epimers, as are D-glucose and D-galactose, which differ at C4. Anomers and mutarotation. Anomers, isomers differing only at the anomeric carbon, which can be hemiacetal or hemiketal carbon. The alpha form of anomer will be called when the OH group is located below the plane of the ring. Here the OH group is present below the plane of the ring, so it is alpha form, alpha D-glucopyranose, and the first carbon is the anomeric carbon. And here the beta form of anomer, when a OH group is located or placed above the plane of the ring, then the structure will be called as beta anomer. Mutarotation, interconversion of both alpha and beta anomers via open chain formation, and it will form the equilibrium mixture. For glucose, there are 36% alpha form, 64% beta form, and less than 1% open chain form. This is the alpha D-glucose, and this is the beta D-glucose, and this is the linear chain. They can interconvert with each of them. Cyclic forms of monosaccharide. Monosaccharides with minimum 5 or 6 carbons, they will form the cyclic structures via hemiacetal or hemiketal formation. Hemiacetal is formed by the reaction of aldehyde and alcohol simply, and hemiketal is also formed by the reaction of ketone and alcohol. When the monosaccharides take a structure of a 6-membered ring, so on the basis of the similarity of the pyranose compound, pyran group, the 6-membered ring form of monosaccharide is called as pyranose, and with the similarity of the furan compound, the 5-membered ring of monosaccharides are called as furanose. The glucose C1 aldehyde reacts with C5OH to form the pyranose ring. But in case of fructose, C2 keto group, here is the diagram. This is the diagram. C2 keto group reacts with the C5 or C6 hydroxyl group, and it will form the pyranose or furanose, and pyranose structure is predominating. Now we know the derivatives of monosaccharides in simple steps: glycosides, sugar acids, sugar alcohols, amino sugar, and deoxy sugar. These are the 5 types of monosaccharides are included in our syllabus. So what is glycosides? Glycosides are formed by condensation of the anomeric OH group with the alcohol. Examples include glucoside, which is formed from glucose, and galactoside, which is formed from galactose. The biological importance include waben, which inhibits the Na plus K plus pump, and streptomycin, which is an antibiotic. So this is the formation of glycosides from alpha D glucopyranose. It will form the glycoside. Sugar acid, oxidation of the carbonyl compounds makes sugar. We know from our earlier chemistry concept. So if oxidation occurs, uh, the oxidation products are following, like aldonic acid. When oxidation of aldehyde group occurs, such as it will form the glucuronic acid from the glucose. Uronic acid, oxidation of the terminal OH group, it will form the uronic acid. Example, glucuronic acid. The first one, aldonic acid, is the gluconic acid, and the uronic acid is the glucuronic acid. Aldaric acid, oxidation of both aldehyde and terminal OH group, glucaric acid. Examples include ascorbic acid. Vitamin C is derived from aldolase. Sugar alcohol, or which is called as alditols, reduction of the carbonyl group may form the polyhydroxy alcohol. Examples include glucose, which will form sorbitol after the conversion into sugar alcohol, and xylose will form the xylitol. They cannot form cyclic structures. These are the diagrams showing the structure of different sugar alcohols. What is amino sugar? One OH group, when replaced with an amino or acetyl amino group, then it is called as amino sugar. It is found in oligosaccharides and polysaccharides. The examples include N-acetyl glucosamine and N-acetyl muramic acid, which is the derivative with lactic acid linked at third carbon. So amino sugar, in case of amino sugar, sugars with an amino group at C2 are amino sugar. They are found in many oligosaccharides and polysaccharides such as beta D glucosamine and beta D galactosamine. Deoxy sugar, when the OH group of the sugar is replaced by hydrogen atom. The examples include 2-deoxy-D-ribose, which is found in the DNA backbone sugar, and L-fucose, which is found in the polysaccharide. In case of deoxy sugars, in deoxy H plus H ion by hydrogen ion replaces an OH, and it will form the deoxy sugar. So my dear students, today we discovered how monosaccharides form the very foundation of carbohydrate biochemistry, from acting as a quick source of energy to serving as structural units and metabolic intermediates. These simple sugars are essential for our life. Glucose, fructose and galactose may look very small, but they drive major physiological processes and build complex biomolecules. Always remember, without monosaccharide, the energy flow and structure of living organisms would collapse. Thank you for watching. Keep learning, stay curious, and I will see you in the next class.